Today, we are at
Pusat Pendidikan Bioteknologi.
Learning Jelebu.
So firstly, let us look into the pregnancy diagnosis in goats. Pregnancy diagnosis in goats using assisted reproductive technologies serves several important purposes, including early confirmation of pregnancy, monitoring embryo development, maximizing reproductive efficiency, and lastly to detect pregnancy complications. For pregnancy diagnosis, we use two types of ultrasound technology, which is first the amplitude mode machine. It is an older ultrasound technology, uses a single sound beam to detect fluid-filled structures, such as the amniotic sac in pregnancy. The second machine is called the brightness mode machine, which is the most commonly used ultrasound mode for pregnancy diagnosis. It produces two-dimensional image by converting returning echoes into varying shades of grey. Each machine consists of two main components, which is first the probe or transducer to emit and receive ultrasound waves. Secondly is the monitor or display screen to convert signals from the probe into real-time image. So the first step in conducting the pregnancy diagnosis in goats is to apply ultrasound gel onto the probe to reduce discomfort or pain and also to improve probe contact. The goat is restrained properly to minimize stress. For the transabdominal method, the probe is placed on the abdomen on the right side near the udder. The probe is adjusted to locate the uterus and detect fluid-filled sacs, fetal structures, or a heartbeat. For the first method, we located the head of the fetus, the heartbeat, and also the vertebrae of the fetus. Next, we use the transrectal method in the ultrasound for pregnancy diagnosis. The same step was done, which is to apply the ultrasound gel onto the probe thoroughly, and the probe is inserted at a 45-degree angle into the rectum of the goat. This is because a straight insertion may capture the bladder instead of the uterus, leading to misinterpretation, and it also helps to prevent damage to the vaginal or cervical tissues. Using this B-mode machine, we did not manage to get clear images of the fetus compared to the first B-mode machine, and this is because the machine is only suitable to monitor early pregnancy, while this goat is already 3 to 4 months pregnant. Therefore, the internal parts of the fetus cannot be distinguished properly. So for this method, we saw the heartbeat of the fetus.
Next is artificial insemination or AI. AI is a breeding method where semen is collected from a high-quality ram and introduced into an old reproductive tract by using a specialized equipment. This technique allows the farmers to improve the genetic traits and increase the breeding efficiency. Now let us go through the equipments needed for the insemination process. First, this is AI gun or insemination pipette. It is a specialized instrument used to deposit the semen into an embryo reproductive tract. Secondly, the straw. This is where the sperm is kept before the inseminating process. Thirdly, the sterile sheath. It is a disposable covers which placed over the AI gun to maintain the hygiene and prevent the contamination. It is also functions to prevent the straw to stick inside the vagina. Fourth, this is called a stopper. Fifth, this is an AI camera. It is functioned to see through the inside EO vagina to inseminate the sperm. Next, this is a speculum. It is a device inserted into the EO vagina to visualize the cervix to accurately inseminate. Let us proceed with the insemination process. It is needed to apply the lubricant to the AI gun for easy insertion and to minimize the discomfort to the yolk. After we insert the endoscope into the yolk vagina, we need to play around slowly and monitor through the camera until we see the cervix. Next, press the AI gun to insert the semen into the yolk's cervix. The sperm insemination is now completed. Easy, right?
Semen collection. For semen collection, artificial vagina were used. It is designed to mimic the natural condition of a female reproductive tract, providing an optimal environment for ejaculation. Warm water was poured inside to mimic the temperature. The actual temperature should be around 38.5 Celsius. The artificial vagina was pumped to mimic the actual vagina, and it is lubricated to mimic the mucus in the actual vagina. Female dummy is needed to stimulate the male. When male started to stand mount, the collector need to redirect the male penis into the artificial vagina to collect the sperm.
Next, we move to the last part, which is the semen assessment. First of all, 20 microliters of the collected sperm by using artificial vagina are pipetted into an appendage tube. Then, a serial dilation is done in which the sperm is then mixed with 180 microliters of extended solution to maintain the sperm viability and to ensure that the sperm sample is at an optimal concentration for microscopic evaluation. The sperm and extended solution is gently mixed by pipetting up and down to ensure the uniform distribution. Once the semen has been properly diluted, a drop of the solution is placed onto a markless chamber in which a specialized counting chamber designed for precise sperm analysis. Then the semen was observed under the light microscope. Now, let's take a look at the result of the observation. The first one is the comparison between two semen samples analyzed under a microscope using a computer-assisted sperm analysis system. The first picture is from KMS group, shows a high motility semen sample, as it can be seen that most sperm cells have long, continuous movement paths, which are represented by the green colored lines. While the second picture is from group 1, represents a low motility semen sample, as we observe a significant number of sperm with shorter movement path, and there are more immobile sperm represented by red colored lines. So, let's take a closer look on the result. As for the concentration, from KMS group, the sperm concentration is lower than the sperm concentration of the group 1. The higher the concentration, it may indicate a denser sperm count. In terms of velocity and progressivity, KMS group sample have higher percentage of progressive sperm as well as higher percentage of rapid moving sperm. This explains why KMS group sample have high motility of sperm. While as for group 1 sample, it has higher percentage of immotile sperm in terms of both progressivity and velocity, so it has a lower motility of sperm. This is because group 1 sample has been taken much earlier than KMS group sample. So, sperm motility decreased over time after ejaculation because it lost their viability and swimming ability. That's all for this practical. Thank you.
Today, I observe, I relate, I share.